Hello and good afternoon. My name is DJ Luloff. I apologize that I could not make it in person for this McNair Symposium. Sadly, I was diagnosed with leukemia at the beginning of this year, and currently at the moment I am on an infusion fighting this disease. If I haven't made it in to speak to you guys today, that is where I'll be. I apologize for my absence. After getting the sad stuff out of the way, let's get into Neanderthals and modern humans, population structures defined by bottlenecks. Does anybody in the audience have Eurasian ancestry? If so, you may also have Neanderthal ancestry. Humans and Neanderthals have mated with each other for at least 300,000 years, with the most important mating events coming around 50,000 years ago. If you look at our image here for human lineage, at about 600,000 years ago, we will see our Neanderthal modern human split from their ancestral population. Humans will stay in Africa, and Neanderthals will move off into Eurasia. The next time they meet will happen around here at about 300,000 years ago. This is the earliest mating event we know of genetically. This map's a little older, so it shows it at around 200,000 years ago, but we know it's closer to 300,000 years ago. As you can see, at about 100,000 years ago, we have these dashed lines going into Eurasia. You may ask, what is this? Well, this is an early migration from modern humans into Eurasia. This is only a temporary migration, whether this is due to a dying off or whether it's due to them moving back into Africa. This is a short-term migration, but did lead to mating between the two populations. And down here, between 65,000 and 47,000 years ago, we see the largest migration, which has the most impact on modern human genetics, as this is where the most recent mating happened between Eurasians and Neanderthals. And with the solid lines, you can see this was a permanent migration where these populations stayed in Eurasia. And you can see with the massive lines going into Africa, this shows that Africans have had much longer genetic diversity, as they've been in Africa for about 300,000 or so years with constant genetic drift. So we would expect them to have the most genetic diversity of the three populations that we are looking at today. An overview for our itinerary. We're looking at Neanderthal and human mating events, effect on population structure. We're looking at bottlenecks' impact on those population structures. And the results, I think, will change the way we look at population genomics. As an introduction to population genetics, it is the study of genetic variance in and between populations, focusing on how allele and genotype frequencies change over time and are impacted by evolutionary forces. Alleles are specific variants of genes, and genotypes are specific combinations of alleles carried in an individual for a particular gene. Alleles can be summed up easiest by looking at hair color. Alleles are a particular set of genes that will always be found in a particular place inside of the genome. The different variants is what makes alleles unique. Alleles will change, like hair color, for example. So it'll be found in the same spot, but the allele pair bond will determine whether you have brown hair, red hair, blonde hair, etc. And we also look at genetic bottlenecks as an evolutionary force. So what are genetic bottlenecks? A genetic bottleneck occurs when a population has a massive loss in population, usually occurring from a catastrophic event in which genetic diversity is greatly limited. After a genetic bottleneck, populations have two options. They can either recover or go extinct. And if you look at our image to our right here, we have a normal population structure, and we can see it goes over at a very high level, and then right when a bottleneck hits, that population collapses. And here's where you can see our two options. They can either go extinct or they can recover. We don't really have great data on whether a population is going to recover or go extinct. This is very environmentally based. A disease could hit, a cold snap could hit. There are many things that could determine whether a population recovers or goes extinct, but this essentially is what a bottleneck is. So what does this have to do with modern human population structures? Great question. So what we were looking for today, and what we were looking for the past few months, we were determined to know what effect bottlenecks have on the modern human and Neanderthal population structures. And are bottlenecks our answer in creating these realistic population structures? So for our methods, we've ran simulations using MS Prime and IPython. MS Prime is used for simulating ancestral histories and DNA sequences in population genetics. We also use effective population sizes. Now, effective population sizes is an idealized population that has no evolutionary forces acting upon them, allowing us to add evolutionary forces like bottlenecks in our research. Our effective population sizes for each populations were as follows. Our Neanderthal population was between 3,400 as a high and 2,400 as a low. This is very recent news. Our research actually got changed greatly due to the Tagore and Akay paper, which came out July 13th of this year, which confirmed the multiple pulse model, which is multiple admixtures or multiple mating events happening between Neanderthals and modern humans. So that's where we get this number for our Neanderthal population. Africans, we have 18,500 for a high and 9,244 for a low. That may seem like an extreme low. This is most likely due to a bottleneck. A bottleneck occurs at about 70,000 years ago, which kills off 99% of one generation of the African population. We are not entirely sure what causes this, but we're pretty sure it has something to do with a volcano that erupted in Africa around 70,000-ish years ago. And then for our Eurasian population, we have 1,500 as a high and 1,000 as a low. This is pretty typical for a founders effect population. We'll come back to what founders effect bottlenecks mean later on in this presentation. But this is typically a small population that breaks off of this African population and tries to explore the New World. And in this case, they moved off into the Middle East and Eurasia. This may seem overwhelming at first, but to the left we can see about 1/32 of our genetic code that we ran. This shows specifically the high population with 2 admixtures and 2 bottlenecks. And to our right, this is what each of these dotted lines are compiling of genetic snippets that we are putting together to make our PCA images. So how population structures are defined, we looked at low and high populations with no admixture, single admixture between the Neanderthal and Eurasian populations, 2 admixtures between the Neanderthal and Eurasian populations, and we looked at 2 admixtures and 2 bottlenecks between the Neanderthal and Eurasian populations. This image to our right is another bottleneck. This will explain our founders effect bottleneck. As you can see, our original population here is getting poured from this new bottle into a tiny jar. And then the bottle will go back and become the original population. What's poured out is representing the population moving into a new environment. If this population then survives, it will become a new population, as you can see by the different colors. This population and the original population are quite different from each other, showing a new population structure has been created. Our results. Well, first we want to state that the African population doesn't change very much throughout these models. We use them as a baseline and a sanity check to compare to the other 2 populations in our models. We ran a no admixture and a single admixture as comparison to the other 2 models. We do know they are inaccurate, especially after the Tagore and Akay paper, which came out on the 13th of July. That really confirmed that 2 admixtures is what happened between the modern humans and Neanderthal populations. And then we ran 2 admixtures with no bottlenecks, and then we ran the same 2 admixtures, but we added 2 bottlenecks between the Eurasian and Neanderthal populations, which shows the Eurasian population crunches and clusters harder than the 2 admixtures with no bottleneck. Before we get started explaining these images, I want to explain what PCAs are. These are PCA scatter plots. PCAs are principal component analyses and is a dimensional reduction technique. This takes high dimensional and variable data such as genetics and reveals clustering of similar observations. It takes complex genes and simplifies them into groups that are closely related genetically. Now, you may ask why we don't use PCA1 and PCA2, because PCA1 and PCA2 show the highest genetic diversity between all of the populations. But the reason we use PCA2, PCA3, which shows the second and third highest genetic variance between the populations is because in PC1, PC2, it shows the separation between the African, Eurasian, and Neanderthals, and we wanted to look at the difference between the difference in the African population, the difference in the Eurasian population, and the difference in the Neanderthal population inside of their own population structures. We didn't really want to look at how far the three populations were apart. We wanted to look at how far apart they were inside of each other, if that makes sense. Figure C and Figure D represents our high and low populations with single admixture. As you can see, we have the African population showing more genetic diversity than our Eurasian population. This is to be expected. The Eurasian population on the high side is a little too close to the African population. That'd be a little less realistic, as Africans have had more time to genetically diverse over the last 300,000 years, where the Eurasians essentially restart their new population, as we saw from the previous bottleneck image. They restart their population around 55-ish thousand years ago when they mate with Neanderthals in Eurasia. Starts a whole new line of sequence. In Figure A and Figure B, this is the high and low population of no admixture. As you can see, same thing. African population shows more diversity than the Neanderthal and Eurasian population. And you'll see throughout the Neanderthal population that very hard clustering, this is all pulled 50 individuals each. So there are 50 individuals in this Neanderthal, in the Eurasian, and in the African population. So you'll be able to tell that the Neanderthal population is in a really nasty bottleneck, and this really shows why they go extinct. In a bottleneck this tight, the limited genetic diversity really hampers their ability to survive as a population. Figure G and Figure H is our two admixtures and two bottlenecks with high and low population. This is our most accurate representation of our population structure, in my opinion, and from what the data shows. We have our African population showing much greater diversity than the Eurasian Neanderthal population. The Neanderthal population, that's 50 individuals stacked on top of each other. This is essentially one individual. This is definitely why they would die off at about 40,000 years ago. The bottleneck that they go through is about 80,000 years in our model and in real life, between 120,000 years to their extinction. They were essentially on life support when we run into them in the Middle East about 65,000-ish years ago. And you can see the Eurasian population really clusters hard once we put it through a bottleneck, as opposed to Figure E and Figure F, which is our two admixtures with no bottleneck. You can see when you don't run these populations through a bottleneck, the Eurasian population has had all of the genetic diversity of the African population, and then just keeps gaining more genetic diversity from the Neanderthals all the way to modern times. With no bottleneck in effect, it almost seems like the Eurasians have more genetic drift time than the African population, which is not what we see. We see the Eurasian creating a whole new population when they move into Eurasia. So this would show that our two admixture, two bottlenecks is the most accurate representation. Bottlenecks are very, very important in creating these realistic population structures to show that these populations don't just run wild. There are evolutionary effects that will slow progression of genetic drift and will increase genetic drift in different scenarios. Well, what are our next steps? The future of our research, we're looking to consider real genetic data. As I may have mentioned earlier, the data we ran with MS Prime is hypothetical genetic data. It's a way for us to run thousands of individuals through some hypothetical models. They act exactly how real genetic data will work. The reason why it's easier to run the theoretical data than it is to run the real data. So if we look at the 1000 Genome Project, the 1000 Genome Project has 2,504 individuals. This is a project that worked for 16 years, between 2000 and 2016, to collect as much genetic data as possible. They got 2,504 individuals from 26 different populations. That is way less than what we used in this model, as you could see by our effective populations earlier in this presentation. Also, we would have to run it against 3 Neanderthals, because that's all we have. We have a Neanderthal from 2022, 2010, and 2012. And I believe not all of them even introgress down to modern populations. They may not even have been part of breeding populations entirely. So we're hoping to try to run these real genetic sequences, since they are public data and we can run them. But I don't know. Most likely we'll have a lot of discrepancies um, that won't be able to be resolved just because we don't have enough individuals. I think for the long term, we're going to have to look at hypothetical data until we can get more individuals, which is coming down the line. This field has only existed for about 25 years in paleogenomics, and it is rapidly going further. I'd like to thank Dr. Allison Mann and Sophia Eberts. I'd like to thank the Mage Lab and the McNair Scholars Program for helping fund this research and questions. Thank you.